Due to DNA sequencing technology, scientists are now able to determine the exact order of base pairing in healthy individuals and in turn identify any mutations that may impair the functional proteins it codes for. The ERBB2 gene is located on the Q arm of chromosome 17. This gene has been sequenced and is found to code for the HER2 receptor or human epidermal growth factor receptor. HER2 proteins are members of the epidermal growth factor family. In healthy individuals, the membrane-bound HER2 receptor binds to growth factors in the extracellular matrix. Growth factors are chemicals that carry growth-regulating orders to the cell by binding to the HER2 receptors, an essential function for growth and division for healthy cells. This image illustrates normal cell division. In cancerous tissue, 25 to 30% of patients are said to have a mutation on the ERBB2 gene. This results in an overexpression of the HER2 protein. These additional receptors will now bind with any growth factors in the extracellular environment until saturated. Cells with an overabundance of HER2 receptors amplify the signaling pathways induced by the binding of growth factors, causing cancerous cell division. Herceptin is one of a group of anti-cancer drugs called monoclonal antibodies. It is an antagonist drug, meaning it binds to the HER2 receptor, blocking access of the growth factor, as a result deactivating the protein and stopping cellular responses. This graph shows the overall survival probability of patients with stage 4 breast cancer. Those patients that were classified as HER2 positive have a much greater survival probability when treated with Herceptin, as illustrated by the green line, versus HER2 positive patients without Herceptin, indicated by the orange line. A greater understanding of our genes due to DNA sequencing technology can greatly benefit mankind. It is astonishing to think that before the creation of the drug Herceptin, women that had the HER2 mutation had the worst survival rates. Now, with the advent of Herceptin, things have reversed. Patients with HER2 now have among the best survival rates.